Okay, so this is a quick review of how prokaryotes replicate. So remember, a prokaryote chromosome is a circular chromosome, and a prokaryote only has one origin of replication. So if I go to my next slide, I have prepared what it would look like in origin replication. So when you have your replication fork opens, we have two replication forks, one on this side and one on the other side as it has opened. So when we looked in class, we were looking at just one side. The picture that I given you from Wikipedia was just looking at replication at one replication fork. So when you have an origin of replication open up, you always have two replication forks. So our leading strand that I'm showing in green is always going to be synthesized continuously as it goes towards the replication fork. So you can see the green strand being made and going in, and as this keeps to open up because of the enzyme gyrase, it's going to keep going on and on. If we look at the other side of the replication fork, if we look on the top side over here, you can see I've also got creating a leading strand going towards the other side of that replication bubble that opened up. So again, this green strand being synthesized continuously, and it will just keep going and going. On the other hand, our lagging strand is not so fortunate because we know that there's a problem with always having to make DNA in the 5' to 3' direction. So you can see these are going to be my little Okazaki fragments that are being made, and they keep needing to be made because this replication fork keeps opening up. And as it keeps opening up, I need to make another strand. And so if we look on the other side, the same thing is happening over here. I needed to synthesize this strand, so there would be my Okazaki fragment going in the 5' to 3' direction. And then as it opens up, I need to come in and make another fragment over here. And it's these Okazaki fragments that are going to need to be joined together by the enzyme DNA ligase. And ultimately, as this replication bubble keeps opening up and opening up, so what you see happening here is on both strands, the new strand is going to be a mixture of lagging strands and leading strands. But ultimately, they're going to be joined together by ligase continuously, so it's going to be one new complete fragment. And so if we go to the next slide, okay, so if we're looking at our newly synthesized fragment, the one that copied off the red template ultimately is going to be a mixture of both leading and lagging strands, all nicely joined in, and the same will be true of the part that came from the blue template. It's going to be a mixture of leading and lagging strands. And so that is what the blue one, the one that copied the original parental blue strand, and the one that copied the original parental red strand. And so you can see we have two new circular chromosomes. And remember, in E. coli, this can happen in about 20 minutes. Prokaryote replication. Oops. So prokaryote replication. One origin of replication. So you always have two replication forks. And so you're going to have new DNA synthesis in both directions until they meet. And the difference being when we look at eukaryotic replication, to a new slide for eukaryotic replication. Remember, a eukaryotic cell has way more DNA, and so if we started replicating at base pair number 1, by the time we get to 3 billion, it would be a long, long time. So eukaryotic replication has many origin of replications. And on each original replication, you're going to have a leading strand going in that direction towards that replication fork, and you would have a leading strand going on the top fragment towards the replication fork. And then as this replication fork is opening, we're going to constantly have lagging strands so that we're always going in the same direction, and these will eventually get joined in. So on this one here, that's going to be my lagging strand on the bottom, but on the top strand I'm going to have a lagging strand going in the other direction. So I hope this helps you understand DNA replication a little bit better and the differences in prokaryotes and eukaryotes. Obviously also in eukaryote this process is happening in that well-enclosed nucleus. In the prokaryote, obviously this just happens right in the cytoplasm. Any other questions? Please let me know.